Well, it started in about 78 or 79. I got melanoma on my left shoulder. It wasn't too deep, so therefore it got cut out. I was told it was extremely dangerous, but after that I was okay. And then in 1993 I got one in the back of my left leg calf. Now it was the size of a ballpoint pen, dot like that on a piece of paper. And then 13 years later, in 2009, I had metastatic melanoma in my body everywhere. And my surgeon, well, I suppose I should go back and simply say our son had passed away 5 weeks earlier from melanoma. Cut me here, and he lifted the bowel out and stripped up the lymphatic system right up to the vena cava, and it had 50% melanoma in it. And there were two over here. When it went off to a branch line over here near the heart and the diaphragm, he couldn't take it. He apologised to me. He said, I could feel them there, but I can't get them. I would have killed you.
Quite often we see that people with melanoma are still quite healthy and living their active lives, but the disease has started to grow and spread, which deems them resistant and no longer able to have immunotherapy. So it's really devastating news when you're healthy and living your life to say, this is it, you're waiting for palliative care now. So I said to him, well, you do know that chemotherapy that has platinum in it damages DNA the same way as UV light. And then he said, tell me more. So I said, we could just pivot and do all our work that we're doing in UV light, but do it on chemotherapy. And that was one of the starts. That was really the start of us then focusing on why chemotherapy doesn't work in melanoma. Which is where we've landed today with the clinical trial. So this study actually started many years ago with us looking at why melanoma patients don't respond to regular chemotherapy. We discovered a process that was silenced or switched off in their tumours, and we used a different chemotherapy drug to switch that on. So in the lab that worked really well, and consequently we started meeting with some of the melanoma medical oncologists, in particular Andre van der Westhuizen. So by using azacitidine and carboplatin, we reset their tumours and got them to start responding to those two drugs. So by responding, I mean they stopped growing. We actually saw for most of the patients that their melanoma tumours just froze in time. They stopped growing, they stopped moving around, they stopped metastasising through their bodies. So by freezing them in time, we could then re-challenge and give them immunotherapy again. And the patients started to respond back on immunotherapy, and we saw a really good outcome. We started off with the very, very first patient that we treated. We didn't have any funding. We didn't have any preliminary data. We didn't have any evidence that the combination of drugs would work. So it was a direct donation by Mrs. Valerie Ryan, who funded that first patient and the first version of the trial, that gave us the information that we needed, that patient's disease did stabilise and it didn't progress. My big dream is for drug repurposing to be used for all diseases. It's not cancer specific, we can use it for anything. And I'd really like for us as HMRI to be the lead site for Australia to start collaborating in some of the international programs for drug repurposing. We have a very unique setup here. We have the clinicians on site, we have most of the diseases that are being focused on being studied in HMRI, and we have the expertise now to do the drug repurposing at scale. So that's what I would really like to see us do.
But the truth of the matter is she's doing something for you and I, which is not just material, and she saves our lives.